Over the last few decades, global male fertility rates have steadily declined, accompanied by a sharp rise in unexplained reproductive failures. When evaluating these issues, the first step in a clinical setting is a standard semen analysis. This test measures three basic metrics: the total count of the sperm, their physical shape, and their forward movement. Relying strictly on these metrics creates a massive diagnostic blind spot. A man can receive perfectly normal results on a standard analysis while still carrying severe, completely invisible genetic defects. As this diagram illustrates, a standard scan misses the internal structure entirely. When we look past the surface, we often find microscopic tears and breaks in the paternal genetic code. This structural damage is known as sperm DNA fragmentation. If a sperm cell with fragmented DNA manages to fertilize an egg, the structural errors disrupt embryonic development. This directly results in unexplained early miscarriages and repeated failed IVF cycles. Solving unexplained infertility requires looking past how a sperm cell swims and examining the actual structural integrity of the payload it carries. Step 1 of the protocol is establishing an accurate baseline. Skip the standard semen analysis and request a specific sperm DNA fragmentation assay from your doctor, such as a Tunal or SCSA test. Once you have your baseline, step 2 involves repairing the underlying biological machinery that causes DNA damage in the first place, the cell's engine. This cross-section shows the biological vulnerability of a sperm cell. It is essentially a fragile genetic payload driven by a mitochondrial engine. Generating the immense thrust required to swim constantly produces metabolic waste in the surrounding fluid. These waste products are called Reactive Oxygen Species, or ROS. If they accumulate in the semen, they attack the outer membrane of the sperm head and fracture the DNA strands inside upon impact. This is where Coenzyme Q10 comes in. Taking a daily dose of 200 to 400 milligrams acts as a mitochondrial spark plug to optimize energy production. Simultaneously, it forms an antioxidant shield that neutralizes these oxygen molecules before they breach the cell wall. Maintaining maximum cellular energy is the most effective way to protect the genetic payload during its hazardous journey to the egg. While energy production handles outside threats, the internal structure requires physical reinforcement. The raw DNA double helix is simply too bulky to fit inside a microscopic sperm head on its own. Step 3 utilizes zinc to solve this. To fit into the genetic vault, the DNA undergoes chromatin condensation. It tightly winds around specialized proteins called protamines to compress its size. A daily dose of 30 to 60 milligrams of elemental zinc acts as the physical armor for this process. It cross-links the proteins, locking the structure firmly together to prevent the DNA from prematurely unzipping when exposed to heat or oxidative stress. However, reinforcing this armor introduces a common failure point, known as the copper trap. This balanced scale highlights the danger. Taking high doses of zinc over several months depletes the body's natural copper stores. You must add 1 to 2 milligrams of copper to your daily intake to prevent a secondary deficiency that will undermine the entire protocol. Dialing in this precise mineral ratio compresses the chromatin securely, preventing the chromosomal abnormalities responsible for early implantation failure. Step 4 focuses on your surroundings. Even the most precise supplementation protocol cannot outwork active, ongoing environmental damage. You need to mitigate external stressors. Keep laptops off your lap. Avoid excessive heat from saunas, minimize exposure to environmental toxins, and prioritize an antioxidant-rich, whole food diet. Step 5 is strict adherence to the timeline. Biology dictates that you cannot alter sperm quality in a matter of weeks. The process of developing a mature sperm cell from a base germ cell takes approximately 90 days. The cell remains highly vulnerable to damage across this entire maturation window. Abandoning the protocol early leaves the rest of the cycle completely unshielded. You must map this execution backward. Start the protocol exactly 3 months prior to your target window for natural conception or a scheduled IVF egg retrieval date. Absolute consistency over those full 90 days is the non-negotiable cost of deep DNA repair. The biological impact of these micronutrients reaches into the epigenetic mapping and lifelong gene expression of the future child. By executing this protocol, you are actively lowering fragmentation and taking direct control of your reproductive health. Pause the video right here and take a screenshot of this checklist. Use it to order your specific labs and dial in your supplements today. Securing your cellular engine and reinforcing your genetic armor is the foundational step toward passing a healthy legacy to the next generation.